I'd like to ask a little bit more about the photodynamic therapy and whether I imagine that there are different dyes with different absorption spectrums and you'd be using different frequencies of light to interact with the
Wavelength.
dyes. Do
Yeah.
you use a whole array of different dyes and wavelengths depending on the purpose?
Yeah. So, I mean, in the field of photodynamic therapy, there's maybe 200 or 300 different dyes have been reported. Now, some of these are derived from natural sources, so things in nature that are deeply colored. So, for instance, your blood is a very deep red, and hemoglobin and has a dye called porphyrin, protoporphyrin, in the hemoglobin. So that's a very good natural dye. Plants and leaves and grass is a very deep green, and that has a dye derived from chlorophyll. Chlorin-type compounds are the green dyes that give chlorophyll its green color. And then a third example is blue-green algae, which again absorbs sunlight. It's a photosynthetic thing, and that has a deep purple dye called a bacteriochlorin. So those are three examples of natural dyes that, if you do the chemistry right, can be photoactive. So it's important to point out that in their natural state, they're not photosensitizers. Okay, chlorophyll absorbs light, it's designed to do that, but it doesn't kill the plant. It synthesizes carbohydrates. But if you do a little chemistry, you can make the green dye from chlorophyll into a very good photosensitizer. Same applies to bacteriochlorins. Hemoglobin is not designed to absorb light, red blood. I mean, it does it very well, but that's not evolutionarily why it was designed. It was designed to transport oxygen. But nevertheless, if you do the chemistry right, you can make very good photosensitizers. Now, a whole range of other ones are synthetic organic compounds that people have designed in the laboratory specifically to act as photosensitizers. And there's a lot of those, hundreds of those even. Some are better than others. It's difficult to use them in the clinics because it's very expensive to do all that toxicology testing and animal testing in order to use them in the clinics. So the ones that are used in the clinics, tend to be being discovered 20, 30 years ago, because really the companies don't have the money to really advance all these new photosensitizers, although most of them are better than the traditional ones. But as you say, each photosensitizer has its own absorption maximum. So it's fairly broad. So you're talking 20, 30 nanometer width of a peak. So a lot of people use lasers to excite them, but you don't need lasers. Same as photobiomodulation, you don't need a laser. LEDs do it very well. And with PDT, people even use filtered lamps. So they would have, like arc lamps, mercury lamps with a filter on. There's a famous example of when they were doing PDT for skin cancer, they used a slide projector, and that was a clinical trial with a slide projector. But nobody has a slide projector anymore.
So how are these sensitizing agents selectively killing malignant cells and leaving the healthy cells alone in photodynamic therapy?
Right. So there's two ways. So one is the dyes, if you inject them into the bloodstream, will selectively accumulate in tumors because of a variety of mechanisms. But probably the most important mechanism is the abnormal blood supply to a tumor, which is leaky and disorganized and tends to accumulate things. There are other mechanisms for injected dyes. For some, like skin cancer, for instance, you generally put the dye on topically onto the tumor. But the important thing is you only shine the light on the tumor. That's a key thing. So, you can focus the light quite well. If the tumor is inside the body, you can often get at it with catheters with fiber optics inside. So you can do that in the lungs, you can do it in the esophagus, in the colon, in the bladder. And sometimes you can even insert needles from outside, put a little fiber optic down the needle. So in principle, you can get at all tumors, even deep inside the body, by delivering the light via fiber optics.
That's really fascinating. Fiber optics seem to be very, very useful in modern technologies like this. You mentioned before that a lot of these sensitizing agents are similar to those found in lots of plant foods, highly pigmented plant foods. So, is it possible to think that if you eat a lot of these pigmented plant foods and you get a lot of full spectrum sun, that you're getting a constant, very low grade level of photodynamic therapy throughout your body?
Probably not. So it is possible to eat things and become photosensitive. And it's generally not good for your skin. The problem is, you see, that the light from the sun doesn't penetrate deep in the body. So if you do eat certain plants, you can get to be photosensitive, but you generally get a bad sunburn. That's what happens. You go out in the sun and you get a bad sunburn. And that is actually a side effect of clinical PDT, that, with some clinical photosensitizers, you have to stay out of the sun for a week after the treatment, or you could get a sunburn.